So if you recall from the beginning of the unit, just after our chemistry review, I introduced the idea of biomolecules. So molecules important in biology and life. And the first one we dealt with was carbohydrates. And just to recap that, the carbohydrates were made up of carbon, hydrogen, and oxygen, and they were made up in a certain ratio, 6 carbon to 12 hydrogen to 6 oxygen. So the second biomolecule we're going to deal with, there's 4 total. The second one we're going to deal with is something called lipids. And this is just a fancy term. You'll more commonly hear lipids called fats. So it's a fancy term for fats most commonly. Okay, and we're going to deal with each biomolecule first in terms of its makeup. So here's the key here. Similarities and differences between them. The similarities is that lipids, so carbohydrates, that lipids are also made up of carbon, hydrogens, and oxygens. But in the end, we need to be able to look at molecules and say, okay, that's a carbohydrate, that's a lipid. And so we need to know what we're looking for when we do that. So even though lipids still contain carbon, hydrogen, and oxygen, the key here is that there's fewer hydroxyl groups. And if you've had a chance to look at the work I left you right before this, was to look up some of the unique properties of water and then define some terms. And 2 of those terms I asked you to define were hydrophobic and insoluble. So remember, anytime you see this word phobic, it means fear of, and water is hydro. So lipids do not like water, and if you think about oil mixing with water, you know that it doesn't mix. And so there's an example of how it's hydrophobic. So they contain fewer alcohol groups than carbohydrates, making them insoluble in water, meaning they do not dissolve in water, they don't mix. Lipids are basically for storing energy. That's their key job in living things, but as well as building some of the membranes. So lipids can be broken down into 4 main categories. We're going to work through each category in order of its importance. So the most common lipid that we're going to deal with is something called a triglyceride. And again, look for key parts of words. So tri stands for 3. And that's exactly what a triglyceride is. It's 3 fatty acid chains attached to a glycerol. And this is the most common one that's found in plants and animals. We can have 2 forms of these glycerides. One can be what we call saturated, meaning that there are only single bonded carbon atoms, and these tend to be solids, and you're going to see why once we have a look at a couple of them. Now, unsaturated fatty acids have 1 or more double bonds in them, so the key to this is the double bonds. And they usually end up being liquid, and the difference between being a liquid and a solid has to do with its chemistry, which is why we spent some time reviewing it. So we're going to come back and revisit why saturated ends up being solid and why unsaturated ends up being a liquid. So next, under this heading, I want to show you how a triglyceride is formed. Now remember at the beginning we talked about 2 types of reactions, anabolic and catabolic, so things that create larger units from smaller subunits, and then the opposite. So what we're going to show you is this reaction in both directions. So we start out with a glycerol molecule. A glycerol is just 3 carbons and 3 hydroxyl groups. And we're going to have 3 fatty acid chains on its own coming in to react with it. Okay, and remember, an acid group here, a carboxylic acid group looks like this, an alcohol or hydroxyl group looks like this. And basically, how we get our fatty acid forming, or our triglyceride, this is where the word triglyceride comes from. Each of these 3 fatty acid chains is going to come in and bond to the hydroxyl group on our glycerol. Now, in order to understand how this happens, I want to bring you back to the 4 types of reactions that we talk about when we're talking about biochemistry. The first 2 reactions we dealt with were hydrolysis and the condensation reactions. So remember, they're the reverse of each other. So if we're going to take glycerol and 3 fatty acids and bring them together, then that's a hydrolysis reaction. If we're going to take that large unit and break it down into its smaller subunits, that's the condensation reaction. So remember, they're the reverse of each other. And the key thing is one uses up water, one breaks down or releases water. So let's just look at this reaction in the forward direction. So we know this is a condensation reaction and 3 waters are coming out. So if you look, for each of these fatty acids coming in, they're going to give up a hydrogen and an oxygen, and the glycerol is going to give up 1 hydrogen off of each stem. Now, if each of these come off, they can form 3 waters. And that's where you get your 3 waters coming out. And remember, once these are removed, the remaining subunits join together and form this bond here, which we call an ester linkage. I don't think we've dealt with this quite yet. But an ester linkage is basically when you have a carbon with a double bonded oxygen bonded to another carbon separated by another oxygen. So carbon, oxygen, carbon, and one of them has a double bonded oxygen on top. That's called an ester link. Now I'm just trying to think of the common questions I usually get in class as well. These R groups here, I'm going to just switch up colors here. These R groups, they're basically just showing you that this carbon chain can continue going and going and going. And instead of drawing out all the carbons that are a part of each of these fatty acid chains, they just use a letter to represent that this chain may or may not be bigger. You could add 10 more carbons, you could add 50 more carbons. It's just showing you that there could be more off of these chains. The important part of these molecules that makes it a fatty acid are these carboxyl groups at the end. So instead of writing out long chains of carbons, they just use letters to represent that. So here and both here. So these actually could end up going depending on what type of triglyceride we're looking at. Okay, and then once we have our larger unit formed from our smaller subunits, sometimes the reverse reaction can happen. So if we removed 3 waters in the forward process to reverse it, we're going to use up 3 waters to give each of these glycerol and 3 fatty acids their hydrogens and oxygens back. So that's how they're just the reverse of each other. Okay, next group under the lipids category is something called phospholipids. And again, try to break down the word to help you know what you're looking for when you're trying to identify it. So phospho is telling you that there's a phosphate in here, okay? And lipids is the fatty acids chain. So the important part of phospholipids, or the important function of phospholipids, is that it's the main component of membranes. We're going to spend a lot more time on this later on, but all you need to know right now is that phospholipids have a phosphate and fatty acid tails, and it's composed of 2 fatty acid tails. So it's always going to look like this. This would be our phosphate head, okay? And the tails that come off of it, there's always just 2. They're going to be your fatty acid chains of tails. Now, just so you guys know what a phosphate head is, phosphate is made up of phosphorus, oxygen, oxygen, and 4 oxygens. And this might be familiar to you if you've had chemistry before. You don't really need to know this structure, but just so you know what we're talking about when we say that the head of a phospholipid has a phosphate group. That's the group they're talking about. Okay, and the last 2 groups we're not going to spend a lot of time on. They're important, but the first 2 were the more important ones of the 2. So steroids is the 3rd group of phospholipids, and it consists of, the key here in remembering is it consists of 4 carbon rings linked together. So when we say that, do you remember when we were talking about carbohydrates, there are 6 carbons rings linked together. These steroids are just considered 4 carbon rings and they're linked together. They're naturally produced in our body and they're necessary for growth. And cholesterol is an example of a steroid, and it's needed for cell membrane function. That's one positive side of cholesterol. Cholesterol can also be bad and clog your arteries. Okay, and then the final group is waxes. Waxes are basically just long chains of fatty acids attached to an alcohol or a carbon ring. They're usually hydrophobic, so they don't mix with water either, and they're firm but pliable. So These are sorts of things when we find a lot on plants, the waxy cuticles on the outside of plants. Even on some types of fruits like apples, you'll see a waxy cuticle on the outside as well. And so that's it for the note on phospholipids. I have attached a video that you're going to watch, and I want you to pay special attention to the chemistry behind why unsaturated versus saturated fatty acids are liquids versus solids, and they do a pretty good job of explaining it, and your textbook deals with it too, but that's really the key thing I want you to take away from this. We have something called an unsaturated fatty acid and a saturated fatty acid, and it has to do with their chemical makeup as to why that is. So pay attention to that in the video, and I hope you enjoy it.